Good afternoon, everyone. It's a great pleasure to be here, and I would like to really thank AMLD for this nice opportunity. So I would like to give you an idea of what we're trying to develop in precision oncology using AI, and what are the challenges that we are currently facing. We now have in oncology several treatments, especially immunotherapies, that can deliver benefit in the long run for a fraction of patients, roughly 20% on that graph. And of course, our challenge is to be able to identify those patients. Who are they? Can we paint them in orange and identify them and only use the correct treatment for those 20, sparing the toxicity and the time loss for the others? Of course, we need to also find pens of other colors to pile up the whole population until we can treat a significant fraction of the patient and derive maximum benefit for them. So what we need for this are predictive biomarkers, quantities that we can measure at the beginning, and that can predict what will be the outcome down the road 5 or 10 years. To do this, we need to interrogate the tumor, but also the microenvironment, and clearly the complexity of that data set calls for AI and machine learning, as our brain are no longer capable of being able to associate some of these features with the outcome. I'd like to show you also how we try to build the evidence. Typically, in a clinical setting, we are going to randomize patients, but first, very importantly, to define selection and exclusion criteria so that this population is minimized in terms of their viability. And in that context, we are extremely performant at running trials and, for example, validating that the experimental treatment here is better than the standard treatment. The problem here is that we do not only bring back this information to the population in the study, but we bring that back to the real-world situation, where oftentimes the patient have a larger spectrum than those of the trial. And basically, we have this dichotomy that we're using results that are not validated in the real-world scenario. So that creates actually an opportunity to actually use digitalization of the healthcare system and interrogate our clinical database directly without this inclusion-exclusion criteria. And basically there, trustworthy AI is absolutely mandatory because we need to be able to mine this information. It will help us, and it's already helping us guide patient treatment. And we, of course, need to have a system that is fair, representative, and of course, trustworthy for the patient. So what are the data types that we're mostly working on in oncology? So clinical data, it goes without saying, more and more patient-reported outcome. The patient can bring a wealth of very useful information that we can start using. Of course, all molecular data, genomics, proteomics, and so on and so forth, and images. I won't have time to detail all these sources, but just to say that's some of the major sources that we are working with. And at the end of the day, what we would like is a decision support tool. I don't think we are at the point here where the doctor can be substituted by an algorithm, but it can be enriched. And clearly, things that we didn't think about or reassure us on a treatment decision are extremely desirable. And in the middle, there is sort of the death valley on how do we go from the left to the right. And there we need a reference data set that allow us to know whether what we are observing is actually meaningful, is it expected, yes or no, and that's a very important aspect to guide decision. And in this gray zone in between, we really need here to build an absolutely trustworthy AI system and data system so that we can go from left to the right. In Switzerland, and I think in many countries, we have made significant progress over the last years as we now have a general consent that is available throughout the whole country. It has been accepted by all ethics committee and all university hospitals and non-university hospitals. So that's very important because now we talk the same language in terms of consent and ethics approval, and that's really a very solid basis to move on. Nevertheless, sharing data still remains to be a very important challenge. And there I would like to show you an example of a program that we have launched 4 years ago called the Swiss Personalized Oncology Program. It's part of the SPHN, Swiss Personalized Health Network. And the idea here is to basically bring the databases of all the 5 university hospitals together to be able to gain in statistical power and to have enough data to train algorithms. We have based all this into the FAIR principle: findable, accessible, interoperable, and reusable. And of course, we really do all our developments to make sure we stick to this principle that are, of course, absolutely needed to move the field forward. Now, on the left-hand side, you see the heterogeneous data set that we have in every single hospital, and it's beyond any hope to try to reassemble that into a coherent data set in between hospitals. On the right side, you would like to really use this mutualized data, and how do you do this transfer? So we had to actually work quite hard with all clinician data scientists to basically build a vocabulary, how we exchange structured data. This has been done and is now up and running and running very, very smoothly. We also have ways to go back and interrogate directly the databases. So in both cases, structured field or actually being able to go to the raw data, we really need a very solid ethic, security, and also an important infrastructure to be able to carry this for the patient. Here is an example of what we had to do. So we had to, of course, go through ethics, do a consortium agreement, a data transfer agreement, a data transfer and processing agreement, and a material transfer agreement. To do this within the 5 university hospitals and beyond really took quite some energy. And so the question is, can we facilitate this process? Are we able to accelerate this through different means? And one solution that came out actually from EPFL, from the lab of Jean-Pierre Hubot, was to use a homomorphic encryption. And the idea is, through this METCO solution that was developed by Jean-Pierre and his team, was to use encryption to be able to securely transfer data across the country. And what we would like to do is to be able to answer a question like, what is the fate of a cancer patient with that mutation, that response to previous line, and then these therapeutic options? And I believe we are very close to being able to do this. The strategy behind this METCO is not a centralized approach, not a pure federated learning, but really a solution where the algorithm goes to the data, but the computation are all done in an encrypted fashion, which means that this is, of course, technically challenging, but this has been done. But the beauty of this is that you can not only use text, structured fields, like I mentioned just a minute ago, but also genomic data. And genomic data can be safely aggregated without ever being in a possibility to see the non-crypted data. Why is that important? It's extremely important for us because that's changing the perception from the governance of the hospital, from the ethics committees, on the way we treat that data. And I think most bodies now agree that this makes us a solution where the data can be considered anonymous. And of course, that's a huge facilitator. I told you the lengthy process of getting those DTUAs in place and the ethics. I think now that we have tools like this, hopefully we are able to really speed up these processes and be able to move in this field forward. You see here an example, a screenshot of what we are trying to work with the group of METCO. We are actually using these ontologies that we have for our program and are able to query the databases and, for example, be able to look at survival outcomes of patients in specific conditions, and that in itself being a big help for the clinic to drive the treatment decisions. So this project has now been taken up by TuneInsight, which is a company that will lend the support for this program, and we really hope to be able to use it in the years to come. So now I would like to show you an example on what we can do with clinical data. On the left side, you see the result of a randomized prospective trial that has been run internationally, actually. And on the right side, you see the data that we are finding in the real-world scenario. And as you can see, we have a fairly good agreement between the trial and the real-world data. This is going back to the first slide I showed you, where we believe mining this data that we have within our hard drives in the hospital might be really a facilitator. And if we are able to reproduce known data, can we interrogate this data set for new questions? And that's an example here. We looked at baseline medication of the patient, and we have seen that patients who have proton pump inhibitors in their treatment when they start immunotherapy appear to be doing worse than those who do not have, and we are linking this to the microbiota that can be affected by the pH of the gut. Proton pump inhibitor will change the pH. We're still in the process of validating this finding, but just to say that trustworthy AI, the fact that we have those big data sets allow us to find hints that could directly link to clinical action, because of course then we would probably prescribe proton pump inhibitor in the patient that will undergo immunotherapy if proven correct. And then to finish up, I'd like to show you an example on how we can use imaging data to move forward. And this is an upcoming field which I believe is extremely promising for precision oncology, called digital pathology. And the idea here is to use the slides, the cuts of tumors that we all have for our patients. You cannot treat a patient in oncology without having this. And to use AI and deep learning to try to associate features of those images to outcome, and of course build a predictive tool. That's an example of a very nice collaboration that we have here with Dorina Tanou and Pascal Frossard at EPFL, where you see on the left side the standard coloration that we do on the slides, H&E. Based on AI, we can identify the different cell types. It's the deep learning part. And then we have sort of this network abstraction of the different cell types interacting with each other. And why we're using this, it's really to be able to build a predictor. You see here on the left panel that the density of lymphocytes that are present in the cut that the pathologist is seeing is not predictive. However, if you then use these network features, I don't have time to detail that feature in particular, you can see that we start to stratify the population between good outcome and poor outcome. And that's really the basis for precision oncology. But of course, all of this really requires AI, and it requires this infrastructure of trustworthy AI to be able to run. Finally, I'd like to touch a word on access equity. We have, of course, thought from the beginning that it's an issue with precision oncology that not everyone gets access to it. And we have here this loop where samples are processed by an expert team, and then the results are sent back to the medical oncologist. But the interest here is that these medical oncologists can be in the university hospital or elsewhere. So we use a Visio system so that anyone basically can send cases to this tumor board. And we believe there are 2 points for equity here. First, access equity. Any patient from the region can get his case being analyzed in this tumor board. And also performance equity. The training is done on every single patient, so we're not here. I'd love to think that we don't have strong biases here, because basically the disease hits everyone, and we are using everyone's data to really build on these predictive algorithms. You can see that we have now quite an activity, around 2,000 patients at the moment, and we're building up on this. So that's the activity of the number of patients we have seen. And examples where this has been highly successful. This patient here was a young patient progressing on a sarcoma. We found that the PD-L1 gene was upregulated, and despite this tumor being notorious for not being sensitive to immunotherapy, the fact that PD-L1, which is an escape for the immune system, was upregulated, prompted us to try an immunotherapy. And you can see that the patient went into a complete response and still doing good now, whereas he was actually progressing very fast on chemo. So to conclude, I think I'll try to show you over the last few slides that I think we have been making some progress in oncology. We believe that precision oncology and addressing the patient to the correct treatment will be a game changer in the years to come, but that clearly AI and machine learning are absolutely mandatory to be able to create this switch. And in this space, clearly, being able to have a healthy, trustworthy AI environment is absolutely critical. We start to have a glimpse on how to do this, but that's certainly something we have to work on very hard in the years to come. So with this, I'll stop and thank you for your attention.